2, 1.
Hello, everyone, and welcome to the Oncomind World Autumn Session. I'm Luca Guayada, Head of Medical Affairs for Thermo Fisher Scientific, and once again, given the unprecedented times that we're living, we are meeting virtually. On behalf of all Thermo Fisher Scientific colleagues, I want to say that we're very happy to offer you again a very interesting and valuable agenda, hopefully for you as well. And so I want to thank you all very much for joining us. So hope you had the chance to see the message from our president, Garrett Hampton, who actually condensed all the passion that we have about oncology testing and also shared some of the plan that we have for the near to come future. If you didn't have the opportunity to check out that video, please, you can do it in the whole of the event. Also, don't forget to check out all our posters with a lot of new data from the Oncomind solution. And for now, as we are already slightly late, and I strongly apologize for that, let me move forward with the first presentation of the part 1 of this event, which is actually coming from Dr. Nicola Normanno, who is Director of Cell Biology and Biotherapy Unit and Translational Research Department at the National Cancer Institute, Fondazione Pascale in Naples. So thank you very much, Dr. Normanno, for joining us. The stage is yours, and I will wait for you right after the presentation for the Q&A session. Thank you again for joining.
Well, let me first thank Thermo Fisher for inviting me to give this talk. These are my disclosure, and this is the pen of my talk. I will start by trying to summarize the current status of precision oncology in solid tumors. Then I'll move to discussion of the ESMO recommendation for NGS testing in solid tumors. And finally, I'll touch upon some open issues. Well, let's start first with what is new now in the precision oncology for solid tumors, and of course, I will start with lung adenocarcinoma. Lung adenocarcinoma is the tumor type in which, for the first time, we were able to identify a number of genomic alterations that offer potential for therapeutic intervention. However, we must recognize that until a few months ago, we had available only a few target therapies for lung adenocarcinoma patients, and basically drugs targeting EGFR mutation, ALK, ROS fusions, and more recently, BRAF mutation. Now, in 2020, we had available a number of very interesting data from clinical trials that have been published or presented in scientific meetings that are going to change really the scenario of treatment of lung adenocarcinoma. And let's start with these two drugs, capmatinib and tepotinib, that showed activity in a patient with non-small cell lung cancer that carried the specific MET alteration, the MET exon 14 skipping, which is found maybe in less than 3% of non-small cell lung cancer. The response rate in patients who had previous treatment is 41% to 46%, and very interestingly, in patients with no previous treatment, 68% for capmatinib. Now, capmatinib has been already approved by FDA, and I think that we will have soon the approvals of tepotinib. RET fusions are found maybe in 1 to 2% of non-small cell lung cancer. Now we have two drugs that will show significant activity in RET fusion positive non-small cell lung cancer, Blue 667, 60% response rate in patients previously treated, and selpercatinib, 64% response rate in patients who had previous treatment. And both these drugs have been approved by FDA. Up to a few years ago, we did not have available any drug to treat KRAS mutant patients. Now we start to have some specific drugs that are active in patients that carry specific KRAS mutation. This is sotorasib, also known as AMG 510, which is active in tumors that carry this specific KRAS G12C mutation, which is quite frequent in non-small cell lung cancer. Up to 13% of non-small cell lung cancer carry this specific mutation. And these are preliminary data showing over 30% response rate in patients that were heavily previously pretreated. And finally, about 2% of non-small cell cancer patients carry HER2 mutation, and in these patients, an over 60% response rate was obtained with this drug that is an immunoconjugate of an antibody directed against HER2 with a cytotoxic drug. Of course, I don't have time to go into the detail of the many clinical trials that are ongoing, but basically this is to show you how the scenario of treatment of lung adenocarcinoma will significantly change in the next few months with a number of new drugs that have been already approved or are going to be approved for treatment of patients that carry specific genetic alteration. Fortunately, this advancement is not limited to lung cancer. There are also other tumor types in which we have very interesting results from target therapy, starting with bile duct cancer, in particular intra-hepatic cholangiocarcinoma, in which a number of genomic alterations that offer potential for therapeutic intervention have been identified: HER2 amplification, BRAF mutation, FGFR fusion, IDH1 and DH2 mutations. We have very interesting data from drugs targeting this genetic alteration, and in particular, one drug, pemigatinib, an FGFR inhibitor, has been approved for the treatment of patients with advanced or metastatic cholangiocarcinoma who carry FGFR fusions. Based on these results, so it's showing an over 35% response rate. And finally, very interesting data are coming also from thyroid cancer. In particular, selpercatinib has shown a very good activity in patients carrying either RET mutation that are usually found in patients with medullary type thyroid cancer, or with FGFR fusion that are found more in papillary and indifferentiated thyroid carcinoma. So basically, what do we expect in the next future? Thanks to all these results of these clinical trials, the fraction of patients who will receive a therapy that is matched to their genomic profile will seem to increase. I mean, in this slide, the circle indicates the current fraction of patients who are receiving a therapy that is matched to their genomic profile, and the arrow is indicated what is the fraction of patients we do expect will be treated with target therapy in the next future. Now, this advancement will be possible thanks to a number of biomarkers and matched drugs that are specific for the different tumor types, as I've shown to you up to now, but also thanks to tumor-agnostic biomarkers and drugs. What are the tumor-agnostic biomarkers? Well, studies of genomic profiling of cancer have revealed some driver alteration can be identified in different tumor types, although maybe at a different frequency. So this led to the hypothesis that a tumor that carries a specific genetic alteration might respond to the matched drugs independently from the histological background. And actually, this has been demonstrated for the first time, I think, for the NTRK fusions. NTRK fusions are found at very high frequency in some rare tumors, but are quite rare in most frequent tumors. However, a couple of drugs, entrectinib and ladroflectinib, have demonstrated that if a tumor carries an NTRK fusion, I mean, the chance to respond to these NTRK inhibitors is identical independently from the histological background. So these are tumor-agnostic drugs, and NTRK fusion is a tumor-agnostic biomarker. Other example of tumor-agnostic biomarkers is the macrosatellite instability, a condition that is quite frequent in some tumors, such as endometrial cancer or colorectal cancer, and more in other tumor types. What this study has shown is that the presence of macrosatellite instability predicts response, this time not to target therapy, but to immunotherapy, to pembrolizumab. And more recently, FDA has also approved tumor mutation burden as biomarker for response to pembrolizumab in patients with solid tumors who received previous treatment based on the findings of this clinical trial. So these data are clearly demonstrating that the number and the complexity of biomarkers that we need to test in our clinical practice, or we will need soon to test in our clinical practice, is rapidly increasing. We have increasing number of genes, and we have increasing complexity of biomarkers, because in addition to point mutation, carbon number alteration, we have also gene fusion, metaxomatic skipping, or more complex biomarkers such as TMB. Now, many of these biomarkers can be assessed by using techniques that are able to test a single gene, a single biomarker by analysis, such as sequencing, PCR, FISH, immunohistochemistry, or it can be assessed by using next generation sequencing that, of course, can assess different types of biomarkers from different genes in a single analysis. Now, the point is that with the increasing number and complexity of biomarkers, it is unlikely that we can continue to use methods to test single biomarkers in our clinical practice. And this is because the cost, the time, and the amount of tissue that we need to use single biomarker tests are not feasible in the clinical practice. And we have already an example that this is already happening, for example, in lung cancer. These are data of 2018. At that time, the coverage for EGFR and ALK testing in non-small cell lung cancer were pretty good, almost 100% in the United States, but the coverage for ROS1 testing was only 15 to 28%, although drugs targeting ROS1 are already available. So it's clear that, because most likely lack of time or lack of tissue, these more rare biomarkers was not tested in the majority of patients. Now, there are some colleagues that still claim that NGS is too expensive, but there are a number of studies, including this study, showing that NGS is cheaper as compared with other testing strategy, at least in non-small cell lung cancer, in which you have a lot of biomarkers already approved, and the NGS is cheaper, also taking into account the fact that if you use a single biomarker testing strategy, you might need to perform additional biopsy in order, to get sufficient tissue to test for all the biomarkers. However, we must acknowledge that in this field there were no recommendations, and this is the reason why ESMO decided to release a recommendation for the use of NGS in their practice. And to do this, what this group did was first to look at the different actionable alteration, list of different actionable alterations that are present in each cancer type, then to perform an escalate ranking for this alteration, and then taking into account only the percentage of patients presenting level 1 alteration, the number of level 1 alteration, and also the complexity of biomarkers, release the recommendation for the use of NGS in the daily practice. But what is ESCAT ranking? What are level 1? Well, ESCAT is the ESMO scale for clinical actionability of molecular targets. Basically, this scale indicates, whether or not there is sufficient evidence from clinical trials, scientific evidence, to use a biomarker and its matched drug in the routine clinical practice. So level 1 are basically the targets that are ready for implementation in routine clinical practice. For level 2, there are the targets that for which we have some level of evidence, but we still need additional data before we move to clinical practice. And then, of course, for level 3, 4, and 5, the level of evidence will be lower. As an example, let's see now the list of genomic alteration according to ESCAT in non-small cell lung cancer. And as shown in this slide, basically, almost all the genetic alterations that I discussed in the first part of my presentation are level 1, EGFR mutation, ALK fusion, METAX for skipping, and again, BRAF, ROS, and TRAC, RET genetic alteration, whereas KRAS and HER2 are still level 2. Of course, this is a dynamic classification. When new data will be available from clinical trials for KRAS and HER2, these two biomarkers hopefully will move soon also to level 1. And the same approach was done for all the other tumor types. This is, for example, cholangiocarcinoma. And as I told you, we have already a number of level 1 genomic alteration, the H1 mutation, FGFR2 fusions, MSI and TRAC fusions. For prostate cancer, we have brachysomatic mutation and MSI, and interesting preliminary data for 10 deletions and mutations. If we move to breast cancer, we have quite a few biomarkers, but some of these biomarkers are quite rare, like intrafusion or MSI, and other biomarkers such as HER2 amplification or PIK3CA mutation can be identified with simple tests like immunohistochemistry or FISH for the amplification and real-time PCR for the few mutations that are targetable in breast cancer. And the same is for colon cancer, which basically KRAS and RAS and BRAF can be addressed by using multiplex PCR, and then transfusions are quite rare. So, I mean, if you want, you can look at this publication in Annals of Oncology. We'll find all the different tables for all the different tumor types. But bottom line, I mean, these are the recommendations of ESMO, and actually these are a recommendation in 3 layers. There are general recommendations for daily practice, there are recommendations for research centers, and then there are some special considerations for patients. In the daily practice, NGS is suggested only in lung adenocarcinoma, prostate cancer, and cholangiocarcinoma. And what is suggested is to use NGS panels that cover only level 1 alteration. If you want to use larger panels, you must be sure that these do not induce incremental cost. And of course, when we think about cost, we do not have to think only to the cost of the panel, but we have to take into consideration also the cost of the drug. If a drug is not approved or is not reimbursed, of course, the cost of the drug would be very high to treat patients outside of the current recommendations. NGS is also somehow suggesting colon cancer, but only if this not leads to additional cost as compared to PCR, and is suggested in ovarian cancer for BRCA1 BRCA2 mutation, of course in carcinoma or non-primary, and in few tumor types for assessment of TMB if a drug is available, if a drug is approved. These are the recommendations for daily practice. However, ESMO recommends for research centers really to use larger panels, because these will improve the possibility to detect genomic alteration that will allow development of patients in clinical trial. And finally, ESMO also acknowledge that there are some cases which the patient and the doctor might decide to use a large panel. But what has to be clear that the advantage from this testing outside of an academic center is very limited, because then the access to the drug is highly limited. So these are the recommendations, but I think that there are few points that still need to be discussed and that are not covered or not completely covered by recommendation. And first of all, there's the problem of the rare mutation, and there's an example of discussed the transfusions. Now, as I told you, transfusions are quite rare in the most frequent tumors, maybe less than 0.5% of colorectal, breast, or non-small cell lung cancer patients carry an transfusion. And actually, what ESMO recommends is that NGS should not be used as a screening method to test for transfusions, because the cost is too high, taking into account the low frequency of the mutation. So cheaper methods are really suggested. However, this recommendation is somehow in contrast with a previous recommendation from ESMO, which basically NGS was suggested only, and immunohistochemistry was suggested only if NGS platform were not available. Well, what is my personal idea is that actually it's very difficult, to use an NGS panel or large NGS panel to screen for NGS mutation, for transfusion, in particular when tumors that have a very low frequency of these fusions. So it's clear that immunohistochemistry at this stage might be a good screening tool, but still I am convinced that NGS should be used to confirm the presence of the transfusions. The advantage of NGS is that NGS will also provide information on the type of fusions, on the partner, and this in the long run would be very important to stratify better our patients and to understand which are the patients that are more likely to respond to treatment. Another tumor that was not recommended for the use of NGS is thyroid cancer, but now with the evidence that RET inhibitors are active in patients that carry RET mutation or RET fusions, I think that NGS should be used at least for detection of the RET fusions. And then there is also an important issue when we will use this panel. Even if you use a small panel, we can have some unexpected finding. For example, if we are looking for FGFR fusions in cholangiocarcinoma, even the smaller panel, NGS panel we will use will include, for example, ARC, ROS, and RET fusion. We can find the ROS fusion in cholangiocarcinoma, and we don't know really how to handle these patients. In addition, even the smaller panel will include a lot of drivers, many drivers, and it's possible we find in the tumor 2 different drivers. This has been, for example, described in non-small cell lung cancer, as shown in this slide, but this is like to occur also in other tumor types. So somehow from testing we can have some complex results, and we know from different studies that the confidence of medical oncologists in using and interpreting results from genomic testing, in particular from NGS panels, is very low. And for this reason, I think that these results should be always discussed within the molecular tumor board. And finally, the ESMO recommendations are only limited to testing of tissue testing in solid tumors. However, we know that there are some patients in which tissue is not available, and we use now liquid biopsy for testing. Now, for liquid biopsy testing, for cell-free DNA testing, we've been using up to now methods based either on quantitative PCR or emulsion PCR because they have a high sensitivity, and whereas NGS had too low sensitivity to be used for liquid biopsy testing. But now we have a number of new methods, new NGS methods that have improved sensitivity and have been developed to test cell-free DNA. And I do believe that when tissue is not available and we need to move to cell-free DNA, we should use NGS as well as we do for the tissue. For example, if in lung cancer patients, we don't have to have tissue available, it's meaningless to test only for EGFR mutation on liquid biopsy basis of the double digital PCR. We should use an NGS also for testing cell-free DNA. So basically, these are my conclusions. I think that really precision oncology is revolutionizing the diagnostic therapeutic approach to cancer, and that the increasing number of tumor-specific, tumor-agnostic biomarkers and matched therapy will lead to always an increase in the need to use NGS for tumor profiling. Now in the current scenario, the use of NGS in the clinical practice should be limited only to selected cancer types as suggested by the ESMO recommendation. But I do believe that in the academic centers, we should continue to perform comprehensive genomic profiling, because only this approach will allow to increase the use of precision oncology, will increase also our knowledge of the mechanism that regulates sensitivity and resistance to target therapies. This was my last slide. Thank you very much for your attention.
Luca, can you hear me? Because I really don't hear you.
Dr. Normanno, I think you should. Are you able to hear us?
No, yes, now.
All right, perfect. So thank you very much for your presentation. Thank you very much. It was very comprehensive. You gave us a very nice overview about the possibility of using NGS. The first question from the audience is, can you tell us a little bit more about combo therapy between targeted therapy and immunocheckpoint inhibitors? How would, for example, testing play a role into this new scenario?
Well, let's say first that the combination of target therapy and immunotherapy are still experimental, at least in some tumor types. For example, lung cancer, we know that if there is a strong oncogenic driver, usually the tumor does not respond to immunotherapy, although now we have some preliminary findings that specific combination patients that have been already treated with CTI might be active also in presence of strong drivers. I think that, the true question here is in many cases whether to go with immunotherapy or target therapy, because for some targets clear that the two approaches are mutually exclusive, but in other cases, we know that both approaches could work. And this is why we need large genomic profiling in order to select the patients that would respond. I'll make an example. For example, in lung cancer, KRAS G12C is predictive response to a couple of inhibitors that are now in clinical development. But usually, KRAS mutant lung cancer is also the tumor type that respond better to immunotherapy. Should we treat these patients first with immunotherapy or target therapy? Most likely, if we have a larger genomic profiling and we go more deeply in the knowledge of mechanism of action of drugs and resistance by accumulating this evidence, we could decide whether these patients should receive first immunotherapy or target therapy. And maybe we can also find subgroup patients in which the two approaches can be combined. We don't have such level of evidence yet.
Yeah, thank you very much for your answer. And now we got a second one from the audience, and this is about a bit more into the direction of liquid biopsy. This is something that we're talking about now since a few years, I would say. But the question is really, it's a little bit general, honestly, as a question, but maybe it is timely appropriated now. And again, the question is, are we finally ready for this now? Or do you think we will keep on, every year we will keep on talking about it, and yet next year is coming, and the next year is coming? Is this really ready for prime time in the routine molecular testing, or are we too far away from that?
Well, it really depends on what you mean to be ready for prime time, because now it is indicated to use liquid biopsy for baseline molecular profiling only in patients that have no available tissue. And in these patients, I can bring my experience to our center. It's almost 2 years that we are using liquid biopsy testing with NGS. And, in the last 4 weeks, we picked up a RAT fusion and a metaxon for Thanksgiving. I cannot say which panel, because, I'm not assuming to disclose this in this session. But, you can find a lot of variants in addition to EGFR mutations when you have no tissue available. Then if we want to talk about the use of NGS liquid biopsy testing to monitor response or for other applications, that's more complex because, of course, in this case, we still need some clinical trials, and we need to prove that really, if we work, for example, with which therapy, we can get some better results. But I think that, more and more we will be using also liquid biopsy for the monitoring of response to patients, at least in selected patient populations, and this will help us to provide a more personalized treatment to each individual patient. But for first line, when you have no tissue, there's no question. You should go with NGS. Don't waste your plasma using a real-time PCR or double digital PCR, because you have at least 5 or 6 targets that are already, for which you have already available drugs, and you cannot look only for EGFR mutation.
That's right. So once again, thank you very much, Dr. Normanno, for joining us today. Thanks for your presentation and also for your answer. We will close now this Q&A and move on with the next speaker, Dr. Giada Rotunno, who is actually a research fellow at the University of Florence. She will be talking about myeloid neoplasma and clinical research around that topic. Please go ahead to the next speaker. Thank you very much.
Hi, everyone. My talk, Next Generation Revolution in Myeloid Neoplasms, focus on how the next generation sequencing in our lab, CREAM, directed by Professor Vannucchi at University of Florence. Just a few slides to introduce you our field of investigation. The classic myeloproliferative neoplasms are clonal disorder initiating by an hit in the hematopoietic stem cells, causing an increased action of mature blood cells of one of the cell lineage in the bone marrow or in the extramedullary sites. The three classic forms are polycythemia vera, PV, essential thrombocythemia, ET, and primary myelofibrosis, PMF. Both PV and ET can evolve into a secondary form of myelofibrosis called post-PV myelofibrosis and post-ET. The three diseases are chronic, but the three can evolve into a secondary form of acute leukemia. MPN are characterized by high complexity, and I try to summarize in this picture. On the left, you can see driver mutations of MEPL, JAK2, and CALR genes. These are defined phenotypic driver mutations since the expression of the mutated genes in cell lines induces cytokine-independent growth, and when expressed in animal models, it resembling phenotype closely myeloproliferative disease. Here you can observe PV is virtually associated with JAK2 mutations, both V617F and exon 12 mutations. While for essential thrombocythemia and myelofibrosis, the mutation profile is more heterogeneous. Mutation also in CALR mutation and MEPL mutation beside JAK2. In these two pathologies, there are few patients, about 10%, that are lacking any of these driver mutations, and they are so-called triple negative. And at the bottom of the figure, you can see in a proportion of these triple negative patients, there are some non-canonic mutations spread in genes MEPL and JAK2 that has been recently described. However, the mutation landscape of myeloproliferative neoplasms is made more complex than initially believed because of the occurrence, particularly among patients with primary myelofibrosis, of additional somatically acquired mutations targeted genes involved in epigenetic regulation, spliceosome, and oncogenes. And these are summarized in the table on the right. Usually, these mutations occur in hematopoietic cell subclones on variable sizes and are so-called subclonal mutations. None of these mutations is exclusive on myeloproliferative neoplasms, since we can find it also in myeloid neoplasms, like in myeloid neoplasms like chronic myeloid leukemia and myelodysplastic syndromes. And these are the World Health Organization criteria for diagnosis in MPN, but this is just to show you how it's important the molecular characterization of this patient, especially when we are in the lack of a clonal marker. So when we are in absence of a driver mutation and we have to study the whole gene, for example, MEPL or JAK2, or if we have to study a more extensive panel of genes, it's important the biological molecular aspect. And these mutations, it's important to remind that these are the already mentioned subclonal mutations of variable sites. And in fact, in this period, from the Italian Society of Hematology expert panel, are listed some recommendation about the appropriateness of the methods for genetic testing in MPN. And for an extended molecular characterization of non-canonic mutations, for example, MEPL or JAK2, and in case of non-driver mutation, where an extended panel has to be investigated, NGS techniques are recommended. So, which is the role of the mutations in MPN? What do they mean and how can we use it? The role in disease pathophysiology and the clinical implications complications, genetic abnormalities occurring in patients with MPN are under active investigations. In particular, it is largely debated what is the significance on these mutations instructing the prognosis, determining the clinical phenotype, and also eventually influencing therapeutic choices. So, clinical research is very important, and it's an active field of investigation, and it's where our group is co-located. This is a translational research project conducted from our group in 2012 in a retrospective sample cohort at the time of diagnosis of primary myelofibrosis. It was one of the most comprehensive studies at the time, since we are talking of Sanger sequencing, both in terms of samples included and of genes investigated. We've studied the molecular asset of almost 1,000 primary fibrosis, and it translated, this means more than 60,000 single PCR. So huge cost, time consuming, but also poor sensitivity to detect low frequency variants. However, the results of this project were very important for the clinical research aspect in myeloproliferative neoplasms, since we demonstrated that testing extended mutation panel, including at least A6L1, IDH1 and 2, EDH2, and SRSF2, is inappropriate for prognostic prediction in selected patients with myeloproliferative neoplasms. We could identify as gene signature that we have defined high molecular risk genes, showed that patients harboring at least one mutation in any of these genes at the time of diagnosis configure a category at high molecular risk. And as you can observe on the left by the graph, the high-risk category showed the negative impact both on overall survival and blast transformation, with respect to patients with no mutation in these genes that were defined as low molecular risk. On the right part of the figure, we show that also the number of mutations in the high-risk genes is a negative prognostic factor in these patients. I show this result just to underline the importance of translational molecular research in this rare hematology disease. So the area of interest of next-generation sequencing in hematology is, as I showed you, of course, myeloid, but also for the lymphoid counterpart. For our area of interest among the myeloid neoplasms, I can mention myeloid lymphoid neoplasm associated with eosinophilia that can show both abnormality of EGFR alpha and FGF, or rearrangement of the T-cell receptor. These forms of MPN are ultra-rare. Until now, fragment analysis by capillary electrophoresis has represented the most common used molecular technique to study these variants of T-cell receptor rearrangement. On the other hand, cytogenetic with FISH and molecular biology with nested PCR are the most used method to study fusion genes. And of course, all these methods represent the limits of sensibility and specificity of the classic molecular biology techniques. So in clonal disease, as hematological neoplasms are, the NGS solution also for these somatic genetic lesions could be very useful. Here, it's clear how the next generation sequencing has grown up in these late 10 years, going from the 2008 when the first NGS sequencer has been placed on the market. And from that moment, the interest of many researchers in the world focused on that. And the publication spread to almost 1,000, less than 1,000 in 2011, to more than 10,000 in the last year. Of course, here I picked up some example of papers in the hematological field, but the number of papers and the publication that I present here is referred to the research of the word next generation sequencing in the PubMed database, so in all areas of science. And now I want to show you our experience with next generation sequencing technique in clinical research in our lab. We were among the first in Italy to buy the Thermo Fisher Scientific first NGS sequencer, the Ion PGM, in the late 2011. And I personally faced NGS technique for the first time with Ion PGM. And that at the beginning, it was not so easy. Of course, being among the first in general was not easy also for the support, because it was always the first time for any kind of problem. But at the end, what we can say is that we grew up together in some way with our field application scientist. So we had the one touch and the enrichment instruments, and it took a bit to have a good manuality with the library preparation and the chip loading procedures, but at the end we've made it. As you can see on the bottom, this is a 316 chip, and it's our best chip loading, manually loading chip. So we were very proud, and we keep it as a memory. But coming back to the NGS, this is one of our first ion ampli-seq custom panel used for clinical research. It was very small, including only 5 oncogenes, 2 primer pools, and 96 amplicons. Of course, we were quite satisfied, because we used Sanger sequencing up to that moment. And instead, this technique allowed us to study up to 25 samples on a single 316 chip. So it was very quick, but of course it strongly depended on the biologic, on the experience of the researcher at the moment, and its manuality. As you can see in the genes included in the panel, these are the same that we have defined high molecular risk. And this instrument helped us to conduct a retrospective study in different cohort of samples to investigate the impact of this signature. After this ion ampli-seq custom panel, of course, we used a lot more comprehensive, including GDS and regions, and we have conducted several research with ion PGM, but later on we decided to go through the new ion S5. And with the new ion S5 Gene Studio, we have definitely improved our laboratory practice with NGS for molecular studies. And this is one of our last custom panel for myeloid genes. It included 27 myeloid genes, almost 300 amplicons, including it included both GDS and hotspots region. But we had a problem with efficiency of some primers, because the results were perfect in silico, but when we performed the first rounds in the lab, we verified that some regions were not covered by the amplicon, or the primers were not performing well as in silico was described. So we were forced to design another primer pool to cover these complex regions, and at the end we had 4 primer pools per sample. These, of course, resulted in higher costs and time consuming for research project that used this gene panel. The S5 was a huge step forward, because it avoided, of course, all the problems related to the initialization with the ion PGM, with the assessment of the pH that was very complicated sometimes. And also the S5 gave us more scalability with the new 530 chip. But this was a faster and more efficient workflow, and of course reduced the cost of sequencing. A small flaw in the system, if we can say so, was the manual loading of the chip for the S5, that in my experience was worse procedure respect to the manual loading of the chip for ion PGM. And at this point, the technology gave us a new possibility. And at that time, we have acquired the Ion Chef in our lab equipment. So it was like a technology revolution in our lab. Because at the moment was the most that we could pretend. It was a semi-automated procedure, quick and reproducible. It avoided the manual loading of the IPS on chip for S5, and coupled with the S5 Gene Studio represented a very improvement for the lab practice. We can say almost the same for the study of the fusion in our research. This is one of our first fusion gene panels that included 51 fusion transcripts. Some of these genes, one, for example, FIP1L1 that we used to study by nested PCR has a very complicated reaction and more than reaction that and with application with next generation sequencing with the AmpliSeq workflow that we were able to do at that time very good. We finally got good results, but also very high detection rate and sensibility for fusion genes. Furthermore, the tool of Ion Reporter allowed us to have precise and reliable results in a very short time. In this case, the upgrade is represented by the commercial Oncomine myeloid research assay. In particular, the panel for RNA can evaluate 29 fusion genes in one single pool and 5 expression genes, and of course 5 genes as control. We actually use it for research for fusion genes, by library prep on Ion Chef or manually, and sequencing on S5, and analysis on Ion Reporter. And coming to nowadays, I would like to share with you our experience with the Oncomine World. We took part of the early access of the myeloid program in the early 2017. At that time, we had not the S5 yet, so we participated at the program with the Ion PGM. So we did the library manually, templating on Ion Chef system, and sequencing on Ion PGM. We have evaluated samples that we already studied in our cohort. So we ran 6 samples of DNA and 6 for RNA on the single 318 chip. And the input material was very small because it required only 20 nanograms of genomic DNA and 10 nanograms of RNA. We had for 30 genes only 2 single pools and a simple AmpliSeq workflow. For the analysis of the sequencing analysis, we have used the Ion Reporter. This was a simple and easy workflow because the sequencing data were sent directly from the PGM to the Ion Reporter. And we had a perfect support from the clinical research assistant consultant, because he was very present and he helped a lot in this process. So later on, after the completion of the project, the new official Oncomine myeloid research assay came to the market, and at the beginning of this year, we began to use it in our research, and this time with library on Chef and sequencing on S5. So these, of course, represent the maximum that we can have at the moment for the lab practice. It's a streamlined process, only 2 days to sequence. And the library on Chef is very easy and simple, and also fast. The only flaw is that you have to work with batch of 8 samples per time, so you cannot multiplex 12 as you could do manually. And this, of course, in one hand, it's more expensive, for sure 30% more than concerning the sequencing costs. But on the other hand, I think that the time that the researchers is saving doing the library on Ion Chef is more precious than money that you could run only 8 samples per run. Moreover, the automation from the beginning of the process guarantees a high quality and standardization of all the process and also of the results. The actual Oncomine includes 40 genes, both CDS and regions, requiring the same small amount of DNA or RNA depending on the application. The analysis on the Ion Reporter is fast, and you can configure the S5 to directly upload sequencing data after the run is finished. And there are frequently updating of the Ion Reporter version, and the support is constantly present for any problem in all the process. Now I want to show you some example of how next-generation sequencing techniques has improved our laboratory practice in some limits that we have found. This is the case of a missense variant in the JAK2 gene. It's a missense variant and it's the most frequent driver mutations in MPN samples. It's a substitution of a G to a T that led to the V617F mutations. It is present also at very variant frequency in the small clones, and actually the gold standard is the real-time PCR. Using next-generation sequencing, we observed a very good concordance between the two methods. Of course, they are not comparable since the real-time is not quantitative, but as you can see in the graph, we observe a very good correlation between variant allele frequency obtained with the oncomine myeloid research assay and the allele burden in the real-time PCR. And as you can see, it's near 1, so the correlation was strong. And in this case, the graph is referred to variant with variant allele frequency less than 2%. In fact, we had a perfect correlation in samples with VAF major than 5%, 100% of concordance for V617F between 5 and 2%, and 98.5% of concordance between 2% and 0.5 variant allele fraction. This is just to tell you that the real-time is still the gold standard for the detection of these variations for diagnostic purposes, but in our opinion, for research purposes, NGS could represent a good alternative to real-time, giving also the possibility to researchers to screen a large number of targets. This is the case of a large indel in the gene carreticulin. The indels of the exon 9 of carreticulin gene have been described in the late 2013 and consists for the most part in large indels. The most frequent is a deletion of 52 base pair, and with our first panel, custom panel, including carreticulin gene, we were not able to call the variant, as you can see in the picture on the left. This is a sample that we already have confirmed with other technique. Sanger sequencing and capillary electrophoresis are the classic molecular techniques used to study this kind of variants. But in our experience, after the failure of the first custom panel, we decided that we needed to design a super amplicon covering the region interested by the deletion. And in the meantime, we decided to switch to use the 400 base pair amplicon chemistry because the length of the deletion was more than 1/3 of the amplicon. And this is a kind of length proportion limit for bioinformatic tools to call a DNA indel. With these changes, we managed to get good call quality with the custom panel. Actually, the oncomine myeloid research assay can call the indel of carreticulin, and we tested the panel with samples that have very low variant allele frequency indel. Also in this case, next-generation sequencing can help researchers to detect DNA variants with more sensibility and specificity respect to standard molecular tests. This is my last example, this time as an insertion of a guanine in a stretch of 8G. This variant is one of the most frequent in the A6L1 gene, and its presence instead of an artifact have been discussed for many years in literature. And also in our group, we decided to study this variant in a retrospective study in a cohort of clinical sample with myelofibrosis. And we confirmed the presence of the variant and also its negative impact on outcome in this sample. As you can observe in the Kaplan-Meier curves on the right, the samples displaying the INSGE in red had worse overall and leukemia-free survival respect to those with other types or no variants in A6L1. But in this case, next-generation sequencing was not useful, and in the figure you can see the hole and the lack of reads in the region interested by the insertion. In the middle, there is the stretch of G. And we had very poor sequencing data, so we decided also in this case to design a super amplicon covering the region to get better result. But We tried both the short amplicon design and also the 400 ones. But also with the oncomine myeloid research assay, we concluded that it's impossible to make the insertion call in this region. So concluding, for this specific insertion in the ASXL1 gene, we'll still have the problem, and we used to perform sanguine sequencing, even if we are aware to lose small clones. In conclusion, I can say that hematological samples are very heterogeneous, and given their molecular complexity, next-generation technologies may become fundamental for the in-depth characterization and study of this kind of samples. Next-generation methods have shown to be useful for translational and clinical research, giving the possibility to study lots of targets on many samples at the same time with very good sensibility. This helps to have rapid and complete molecular information about the samples in the study. And regarding the more technical aspects, I can say that the oncomine myeloid research assay, coupled with Ion Chef doing the library on Chef and templating on Chef, is a streamlined workflow. It's the fastest solution that we have in the lab at the moment. Of course, in this period, a new possibility has faced on the market with the GenXus, and probably we will think about it in the future. The possibility to have the Ion Reporter software tool dedicated to this specific panel, it's very useful. Ion Reporter is continuously updated with the aim to give back analysis of the data even more clean and specific. And finally, the support by the clinical application research consultant is fundamental, and it's always present. And thank you for your attention.
And thank you very much again, Dr. Rotunno, for your presentation. Very, very interesting, comprehensive, and also I appreciate very much you sharing your very personal experience using first the PGM and then moving on with a higher level of automatization with the Ion Chef and the S5. So again, thank you very much for that very practical experience. So now we will move on now to the question for your presentation. And the first one is about, in general, the role of the NGS in hematological disease. Now, you have shown some very peculiar application, meaning that you have shown how you're using NGS for some very specific disease. But the question here is about, do you see in the near to come future the application of NGS also expanding outside the hematological disease that you have just shown? So in other words, do you see a much higher uptake in the general hem-onc field of NGS, or do you still see it very well defined only to some very specific diseases?
Okay, thank you for the question. I think that with the new technique for study by molecular biology, we will have even more great number of genes for many hematological disease. We started, like as you said, with a small panel in our experience, and then in a few years we moved to a very extended panel. I think that this possibility can, of course, be extended also to other kind of disease instead of only myeloproliferative myeloid, of course, because these new molecular techniques like NGS are let you study a lot of target per time, and then when you go for fishing research, as we used to call, like when you, for example, discover a new gene implicated, it's more easy with NGS to study a lot of genes in the same run, so in the same experiment. So I think, yes, for sure, NGS can be useful also for other diseases, yes.
All right, thank you very much for that. Now, another question for you, it's actually, let's say, continuing of your story. You have moved from PGM, which was honestly a very complicated instrument for a number of reasons. It required a lot of manual working, and then you moved on to the Ion Chef S5. And that's yet another opportunity now with the new Ion Torrent Genexus system. How do you see, in your experience, the importance of having high level of automatization? So what is the real benefit that you have seen, again, passing from the PGM to the S5? Have you seen less error? Have you gained, have you been able to reduce the turnaround time? What was your main advantage for you?
Yes, of course. The turnaround times is, of course, reduced, drastically reduced. The hands-on time of the researcher is, of course, reduced because, of course, you can do more research respect to manual preparation and operation in the lab. So you can evaluate more aspects and you have more time to doing real research. Of course, there is a standardization higher than with the manual preparation of the library, and the data are clean and more reliable. Yes, I think this is the automatization is very useful. We don't have Genexus, as I told you, but of course it's a possibility that we are evaluating for the future, because of course molecular biology and NGS techniques are taking even more space in our research. So of course automatization is the first the preferred procedures in our lab. Yes.
All right. Thank you very much again also for this question. And now another one which is coming in the direction of MRD, minimal residual disease monitoring. That's a very hot topic at the moment. It's only used in certain hem-onc, especially, in the context of ILL. So the question is, again, how do you see now minimal residual disease expanding also outside, let's say, the classical nowadays implication? And also, do you foresee there's going to be an increase in terms of a burden from the laboratory that are involved in MRD, because there's going to be many more cases in which people will want to use MRD as a tool to assess, patient, let's say, progression?
Yes, of course, yes. This is a very hot topic at the moment, also because, there are some tricky points with the NGS analysis. For example, how in depth you can do with the VAF, for example. But I think that in our experience, we can evaluate a sort of MRD with NGS also for hematological disease. For example, we have conducted a study, a retrospective study on samples after the transplantation, the bone marrow transplantation, and we observed that a real good, I can see, a real good correlation with the variant present at the moment before the transplant and then after the transplant, and when the disease started again, and so we can evaluate also with NGS the clones moving in the in the disease. So I think that it's a hot topic, but of course you have to be very sure and very practiced with the interpretation of data, and also with the standardization of your technique, because of course there is the problem of the false positive, the technique problems, and so I think it's important to have a very good standardization workflow in the lab before doing this, because there are a lot of helps from the from the company, from the kit, but you have to take very careful the data that you have. You have to be really sure to do MRD with NGS, I think, but it's possible.
All right. Thank you again also for this answer. So now, Giada, I'm going to close the Q&A. I would like to thank you once again for joining us as a speaker in this event. Really excellent presentation, and also thanks for taking the time to answer to those questions. So now to everyone, we're going to have a break of about 10 minutes because we were slightly late today. Please take the opportunity to go get a coffee and also have the chance to navigate the event by looking at the presentation hall and the poster hall. And we will be back here for the next presentation in about 10 minutes. Once again, thank you all very much for joining us today.
Thank you, Luca.
Bye, Giada. Ciao.
Bye.